as well. So hopefully we don't experience that. But let's see.
And I'm back. Good morning, good afternoon, good evening, AP Biology. And
so per usual, I do want to just watch it the first time. We'll try to, don't even worry about writing anything down this first time. Let's just watch and listen, and then I'll go back and point out the things that you should be writing down.
Welcome to AP Biology Daily. This is topic 3.6, cellular respiration. And this is Daily Video 1. My name is Chris Mansour, and I'm coming to you from room 102 at Tiffin Columbia High School in beautiful downtown Tiffin, Ohio. In today's Daily Video 1, this lesson will describe the process that allow organisms to use energy stored in biological molecules. So now we're going to focus on us. It's all about us now, how we use energy. And the skill that we're going to focus on is constructing a graph, plot, or a chart, which is very important. And so what will we learn? We're going to learn a lot of good stuff. But first, what processes do all forms of life use to produce ATP? How do eukaryotes coordinate cellular respiration? And how is an electrochemical proton gradient formed during cell respiration? Again, might be a good little pre-assessment to pause the video and try to jot down some answers. So, fermentation and cell respiration are processes that allow organisms to use energy stored in biological molecules. You may have heard of these terms before. And so, cellular respiration and fermentation are characteristics of all life forms. All living things have to use energy in one form or another. Cellular respiration and fermentation release chemical energy from organic molecules like glucose. As you see in my image there, we have glucose as the starting point. And so oxygen, though, is not used during the process of fermentation, but it is used during the process of cellular respiration. So if you look on my image there, you can see where I put a red box around oxygen is not present. And then oxygen is present. So it's kind of like you get to a fork in the road. Oxygen not present, oxygen present. Which way do we go? And the one key thing here is that fermentation and anaerobic respiration are not the same process. Fermentation, the products are lactic acid or ethanol, where in anaerobic respiration, we still have pyruvate. So that's just kind of a key point I wanted to point out there. So again, what I tell my students is draw the pictures. So maybe you may want to pause the video here, draw this picture, maybe add notes to it as we go. Again, use these videos as you see fit. So let's talk about cell respiration a little bit. Cell respiration, eukaryotes, we're eukaryotes, right? So we're doing cell respiration now. Involves a series of coordinated enzyme-catalyzed reactions that capture energy from biological molecules. There's a lot of words in that sentence. Maybe if there's some terms you're not sure of, pause the video and define them. So I have a picture here of cell respiration, a quick overview. Again, maybe stop and draw, may be helpful. So cellular respiration involves the release of chemical energy through the breakdown of glucose and creates an energy storing molecule called ATP. Again, ATP is ultimately what we want to produce. And so what cell respiration does, it takes this chemical energy through the breakdown of glucose to create more ATP. So ATP is used by all cells to do biological work. So biological work to transport materials, for movement, for reproduction, whatever it may be, ATP is used by cells to do work. And cell respiration involves multiple metabolic pathways. We're back to those pathways again. Pathways are important, right? They're sequential so that you can release small amounts of energy each time. And so our first one is glycolysis, which occurs in the cytoplasm. So you can see where that arrow just popped in. That's glycolysis. all of these processes start with glycolysis. Glycolysis is a conserved process. All living things do glycolysis. Pyruvate oxidation. All right, pyruvate, right, which is one of the products of a reaction in oxidation. You're losing electron. That occurs in the mitochondria. So you can kind of see where I put that red bracket in. And then there's the arrow pointing to that process. The Krebs cycle, also known as the citric acid cycle, occurs in the mitochondria. And so there's your Krebs cycle, also known as the citric acid cycle. And then finally, we have the electron transport chain, which occurs in the mitochondria. So again, a lot of steps here, a lot of small pathways. I would stop, draw the picture, put glycolysis, pyruvate oxidation, Krebs cycle, electron transport. And so let's talk about electron transport first. So electron transport chain transfers energy from electrons in a series of coupled reactions. And so essentially what the electron transport chain does, the reactions occur in membranes of chloroplasts and mitochondria and in the cell membranes of prokaryotes. And so there again, in that blue box, I'm showing you electron transport chain. So an electron transport chain facilitates, again, facilitate means to help a series of coupled reactions used during cell respiration. Again, if you're not sure what coupled means, maybe pause the video, maybe ask a friend, put a text in the Zoom chat. And so the electron transport chains allows for more controlled and efficient transfer of energy. When I talked about those biological pathways before, you want these small series of steps. It's more efficient. And then the electron transport chain uses electron energy to establish, again, an electrochemical proton gradient across the membrane. Lots of words. Again, don't get overwhelmed. Pause the video, draw the picture, go through and figure out ways to define these. But the point of the electron transport chain is to create this electrochemical proton gradient. And there it is. There's that creating that gradient as those protons are moving across. And so what will happen is those electrons are delivered by electron carriers called NADH and FADH2 to the electron transport chain. So again, lots of information here. Break it down into smaller steps. But in that green box down there, you can see where these carrier molecules come in. So again, pause and draw, you know, kind of label these pictures because there's a lot going on. And then finally, in this step, ATP synthase, again, that enzyme, we know it's an enzyme because it ends in ACE, uses the electrochemical proton gradient to synthesize ATP. So ultimately, we're making some ATP. So again, a lot going on in this picture. Pause, draw it, do your own colors, label it. But again, I think we learn really well from drawing out these processes. So let's practice a little bit. There was a lot there. This is going to, so in this lesson skill, it's representing and describing data. We're going to construct a graph, plot, or chart. And often you have to be able to interpret graphs and charts, or often you have to draw them. So here is our data. We're going to representing and describing data. Yeast cells are placed in an apparatus with solution of sugar, which is a major nutrient for yeast metabolism. The apparatus detects bubbles of gas released by yeast cells. The rate of respiration varies with the surrounding temperature as indicated by the data below. So you have temperature, you have number of bubbles of gas. I'm not going to go through and read those for you. Pause the video. Graph the results represented in this data table. So this is where I would pause, do your graph, because in the next slide, I'm going to give you the answer. So go pause the video, draw out your graph. You know, if you're doing this in class, hold them up, show your friends. Maybe you need to put something on the refrigerator for mom and dad or whoever you live with. All right. So here is what a graph which would be acceptable, or which is very important.
Okay, so this is a lot, and I think what I've learned not only in the last couple of weeks with you all, but also last year with my other AP biology students is that focusing on something as specific as what he just went through is not always going to be the most helpful, because you all need to know these general concepts. You need to be able to pick out the words. What they'll do on the AP exam is they're going to give you some hint words. They'll use words like electron transport chain. They'll use words like ATP synthase. And you don't need to know exactly when this is happening or exactly the mechanisms that allow this to happen, but you should know in general what's happening in these processes. The electron transport chain just means that electrons are being provided by one molecule, they're being moved throughout a membrane, and along the way, we're using their energy. ATP synthase just means that we're using an enzyme to synthesize ATP. So as long as you all can keep up with those general bang for your buck concepts, I think we're going to be in a good place. Sometimes when you get so dug deep into the specifics, it just all gets too jumbled and you don't make the larger connections that you all need to be making. And so that's what I want to point out here as we go over the images. I do want you, however, to write down these questions. So I'm going to leave this on the screen for 30 seconds or so. I do want you to write down these questions because they allow you to kind of think about everything that was said. And then as you return to your notes, you know which questions were answered in this video. And let me know when you're done writing these 3 questions down.
I'm done.
Thank you.
I'm almost done.
Okay, so now I'm going to move on. Now,
Okay.
okay. So I do think it's a good idea to draw this image. This is a very simplified image. I think you all, for the most part, have heard these terms before. Maybe you're not so familiar with the term glycolysis or the term pyruvate, but you've heard some of these other terms before. So this whole process of cellular respiration is what we consider a conserved process, which means that it has been conserved through all forms of life. We see this appearing evolutionarily across all different species. We have not yet come across a form of life that does not need to undergo this process of cellular respiration. The reason for this is because all forms of life need energy. So as you all draw this picture, actually, I don't know. I think that's basically all you need to write down from this slide is just this image on the right. So once you get this general flow chart, we can go ahead and move on. So again, let me know when you're done drawing that image there.
I'm done drawing.
Thank you.
I'm done.
Okay. Okay, so this image I will not have you draw, but let's just talk about it a little bit. So first of all, it says cellular respiration in eukaryotes involves a series of coordinated enzyme-catalyzed reactions that capture energy from biological macromolecules. Let's break that down. What is a eukaryote? What is a eukaryote? Hello? What do you know what a eukaryote is, Lexi? Or a eukaryotic cell?
Oh, isn't it? Yes, it's the more I mean, not the more chaotic. It's the more like complex cell.
And what
It's
makes it more? I'm sorry, I didn't mean to cut you off. What'd you say?
it's younger.
Yes, it's younger. Yeah, evolutionarily, it's younger. And I love that you said it's more. Did you say more? Yeah, you said more complex. What do you mean by more complex?
It has membrane-bound organelles, right?
Excellent. And what is the probably the most important membrane-bound organelle?
Nucleus.
Excellent. Yeah. So these eukaryotes are, for example, animals and plants. They are not bacteria. Bacteria do not have nuclei. But good job, Lexi. You're 100% right. They have membrane-bound organelles. They have what's called compartmentalization. So the cellular respiration is taking place in eukaryotes or eukaryotic cells, and it involves a series of coordinated enzyme-catalyzed reactions. What does that phrase mean? Coordinated enzyme-catalyzed reactions. What does again, let's just break it down. When we see these phrases, these big terms, I know we know what the individual words mean, so let's just break it down. What does it mean for something to be coordinated?
To be like together in sync, kind of.
Yes, exactly. To be together in sync. That's a great way of putting it. Or we could say maybe to be in sequence with one another, like a dance routine. What about enzyme? What is an enzyme?
What did you say? What was the last part?
What is an enzyme?
Energy? No, no, no.
So protein.
It is a protein, and what does it do? I'll give you a hint. It's the word that is right next to the word enzyme.
It's a protein, right?
Yes. And go ahead, Selena.
Doesn't it speed up reactions?
Exactly. So enzymes are a specific class of proteins that catalyze things. They speed up reactions. So now we put it all together. A coordinated enzyme catalyzed reaction, or multiple reactions, is as Selena said, a series of reactions that are in sync. And then as you both came together to say, those series of reactions are sped up by enzymes, sped up by a specific protein called an enzyme, or multiple enzymes. Okay, so that's what we mean by coordinated enzyme catalyzed reactions. Now, if you think back to a few weeks ago, we talked about the fact that instead of chemicals going from A to D, which would be a huge jump, it will take a long time and it will take a lot of energy. What they'll do is they'll split it up to smaller steps. They'll go from A to B, then from B to C, and then from C to D. Or maybe they'll split it up even further, go from A1 to A2 to A3 to B1 to B2 to C to D. Okay, so they split it up, and that's what we're talking about here. They're coordinated enzyme catalyzed reactions. This helps with the energy efficiency of the process rather than having to put in a whole bunch of energy to go from A to D, right? Just imagine the amount of energy it would require for you to jump from ground floor to fifth floor. You'd have to be the strongest person in the history of human evolution. But to go up a flight of stairs from the first floor to the second floor wouldn't be that difficult at all. Or to walk up, let's just walk up a slight incline. It might take you a long time, but you could do it pretty easily. And then we throw in, let's say we put you on a slight incline and you're eventually going to go that entire height of the 5 floors, but we give you a bike so you can ride your bike up this slight incline. So not only is it easier, it doesn't require as much energy, but you're able to do it faster. But that's what we're talking about here. Instead of doing it all at once, we're going to take several smaller reactions. We're going to pair them next to each other, and we're going to use enzymes to speed them up. Okay. Now, in this case, the reactions that we're talking about are listed down there at the bottom of the slide, and I do want you all to write those 4 reactions down. They are referred to as the metabolic pathways. The first one is glycolysis. So instead, what I would ask you all to do, instead of bullet points, why don't we write number?
The picture.
Sorry.
Put glycolysis.
Let's write numbers. 1, 2, 3, 4. 3 of the 4 are taking place inside of a mitochondrion. The first one, glycolysis, happens inside of the cell's cytoplasm. Okay, now one thing that, I kind of wish we had an AP chemistry class at West Mac, but we teach this concept in AP chemistry, but it's also relevant to AP biology. This process of oxidation, it says pyruvate oxidation. Oxidation means the process by which a molecule loses an electron. The opposite of oxidation is called reduction. And reduction is the process by which a molecule gains an electron. And I just sent both of those definitions to the chat. I would write that somewhere on the side. We do see this term come up a lot, especially oxidation. We see it come up a lot because we need electrons for the electron transport chain. So they have to come from somewhere. And we, in this case, get them from pyruvate oxidation. Where did the electrons come from right before, at the beginning of the light-dependent reactions? At the beginning of photosynthesis, where did those electrons come from? Does anybody remember, where do we get the electrons at the beginning of the light-dependent reaction? It comes from something with which we're probably all very familiar. You probably drink some every day.
Water?
Water, yeah. Do you remember we talked about how water molecules get split? And that provides an electron. But my AP chemistry teacher when I was in high school helped me to remember oxidation and reduction with that saying that I just sent to the chat. Leo the lion goes grr. Leo is loss of electrons is oxidation. and ger is gain of electrons is reduction. Leo goes ger. It's always good to have some mnemonic devices in your back pocket to help you remember these difficult scientific questions. Let me know when you're done writing these bullet points here.
I'm done.
Great. Selena, what about you?
What you say?
I said, let me know when you're done writing those bullet points.
I've been done.
Oh. Oh, shoot. Let me point out one more thing on this slide. So we can see, you know, this is a good image. I just don't want you guys to take time drawing this. But we can see going into the cell is food. That's where we ultimately get the glucose, most of us. Now, plants obviously are not eating food, so we know that they get their food from photosynthesis. We talked about photosynthesis. We saw how at the end of the Calvin cycle, glucose is produced. So that's ultimately what we are talking about when we say food. We're talking about glucose. We also see that oxygen is diffusing into the cell. Both of these are reactants in cellular respiration. Cellular respiration is an aerobic process. Sending that to the chat now too. The word aerobic means that it requires oxygen. And then if we look closely, we can also see in this image carbon dioxide leaving the mitochondrion. We can see water leaving the mitochondrion. And in addition, we can see in several places ATP is produced. At the end of glycolysis, ATP is produced. At the end of the Krebs cycle, ATP is produced. And at the end of the electron transport chain, we've got ATP produced again. So this process of cellular respiration will produce about 36 ATP molecules. It's really good at what it does. One more question here on this page. And the answer is also written on the page. So as I ask the question, I want you all to kind of scan the words that are on the page because the answer is there. What does glycolysis mean? What does this word glycolysis mean?
I mean, it says something that occurs in the cytoplasm on the page.
Okay, it is a process that occurs in the cytoplasm. That's true. What else? What else can we say about it? Keep looking.
It looked like it turned into ATP. Is that what that is? The arrow's pointing towards it.
Okay, so yes, the process of glycolysis does produce ATP. What word does glycolysis sound like? The first part of it, at least. Selena.
I don't want to say the wrong answer.
It's okay if you're wrong.
Glucose.
Yeah, glucose. So we've got that is what's meant by glyco. We're talking about glucose here. What about lysis. Does anybody remember? Lexi, you were really good at this. This was, I think, in our first unit. We talked about lyse. When something lyses, what does that mean? Lyse.
Could you give me a hint?
What happens when too much water flows into a cell?
Oh, it explodes.
It explodes, it bursts, or it splits. That's what lyse means. So I'm sending a lot to the chat right now. But I hope you guys are writing these messages down. So lyse or lysis, they both mean to split, to break, to burst. So in this case, what we're talking about is glucose splitting. So the process of splitting glucose apart releases this ATP. That's what glycolysis is. Glycolysis occurs in the cytoplasm, and it means to split glucose, to ultimately make ATP. That's what our goal is in these processes, to make ATP. So actually, let me add one more thing. Glycolysis, I can't spell. We've got these 4 processes that we were talking about. Number 1 is glycolysis. Number 2 is pyruvate oxidation. Number 3 is the Krebs cycle. And number 4 is the electron transport chain. Okay. So 3 of the 4 processes have the goal of making ATP. Pyruvate oxidation is meant to release an electron, which you need, again, to make ATP. So that is the overarching goal here. We're trying to make ATP. We're trying to make energy. So again, I do want you all to try to write down these notes that I'm sending to the chat. We can continue to go over them, but I want you to have them. Okay, let's keep going.
So again,
so just as in the last slide, we talked about these coupled reactions. We talked about these coordinated enzyme-catalyzed reactions. A series of coupled reactions just takes one reaction and puts it next to the other, so that the products of that first reaction become the reactants of the second reaction. That way, nothing is wasted. This is an extremely energy efficient process. And this is very similar to what we talked about during the light dependent reactions. What we're going to do is build up, if we look inside of this red box here, and you guys don't need to draw this image, but if we look inside of this red box here, we're building up a proton gradient. So this bullet point, this 4th bullet point, electron transport chains use electron energy to establish electrochemical/proton gradients across membranes. This is your bang for the buck bullet point here. So let's break it down. Electron transport chains are processes that do exactly that. They transport electrons. They take electrons from one source and they move them throughout the membrane typically. In this case, the membrane is, well, in a eukaryote, it would be the membrane of a chloroplast or a mitochondria. But for prokaryotes, they don't have chloroplasts or mitochondria because those are membrane-bound organelles. So this process of electron transport takes place inside of their cell membranes, but it's always going to happen in a membrane. That's good to know. So electron transport chains use electron energy. They use electron energy. So what they're going to do is excite electrons. They're going to provide energy to them in some way. We know that from photosynthesis, most of that energy is coming from sunlight. That sunlight is being used to excite the electron. But in this process, something a little bit different is going on, and we'll talk about it. But they're going to use the electrons, and as those electrons return back down to their ground state, they're losing energy. They're losing energy. And I put losing in quotes because we know energy is never fully lost. It cannot be destroyed. So when the electron loses its energy, that energy is not actually just disappearing. It's being used to do some type of work. It's being used to move something from one place to the next. So electron transport chains, which are taking place in membranes, use electron energy to establish electrochemical proton gradients. All that means is that they're going to concentrate protons, which are hydrogen atoms. They're going to concentrate these hydrogen atoms somewhere on the inside or on the outside of the cell. Do we see how these red arrows represent the movement of hydrogen atoms? They're moving through these big proteins inside of the membrane. So what happens, and Lexi, you've kind of been consistent in giving us this answer, but what happens when we end up with a whole lot of, what's going to happen when we end up with a whole lot of hydrogen on this side of the membrane?
It's going to move to the other side.
It's going to move to the other side, yeah. And so once we get a whole bunch of protons, and I'll type that into the chat. Protons are H plus ions, hydrogen ion, positively charged hydrogen ions. That's what we mean when we say a proton in this case, a hydrogen ion. Once we get a whole bunch of those protons on one side of the cell, naturally they are going to diffuse back onto the other side of the cell. That's going to be a spontaneous process. We don't need to use energy to do that. So hydrogen is going to flow through this ATP synthase, which of course makes ATP. It does phosphorylation. So this is the process of oxidative phosphorylation. Oxidative, I think it's with an I actually. Using energy from electrons to combine ADP with a phosphate group, making ATP. So again, this seems like a lot, and I really, really don't want to get too deep down into the weeds here. I don't want to get too deep down into the weeds because some of this stuff is just not going to provide you with much insight that you can use on your AP exam. But you do need to be able to, in general, recognize these processes. So when we say oxidative, again, I need you guys to recognize that what we're talking about is something losing electrons. That's what oxidation means, to lose an electron. When we see phosphorylation, I need you guys to start to recognize that we're talking about taking an ADP, so two phosphates, combining it with another phosphate group to make ATP, adenosine triphosphate, three phosphate groups. So these words, when we start to make the connections, are going to be so much more important to you than knowing, okay, so it's going to move through protein complex 1, it's going to, then the electron is going to be transferred to this kinokine, then it's going to, we don't need to know all that for this class. This is not a graduate biology class. You do, however, need to know these basic terms. You do need to know what phosphorylation means. You do need to know what an electron transport chain means. Okay. So, and I'm going to probably make my own notes for this so that we can break it down even further, and then we're going to do a virtual lab that might help as well on Friday. Let's just take one quick look at this. Yeast cells are placed in an apparatus with a solution of sugar, a major nutrient for yeast metabolism. The apparatus detects bubbles of gas released by the yeast cells. The rate of respiration varies with the surrounding temperatures as indicated by the data below. So graph the results represented in this data table. This would be, extremely simple to graph. Which one should go on the x-axis here? Temperature or number of bubbles? Which one should go on the x-axis?
Temperature.
Good, yes. And temperature is your independent variable here. So of course it should go on the x-axis. Then we're going to put number of bubbles on the y-axis. Now we've got a pretty good title here as well. So again, I always want you guys to think about the TAILS acronym. We need a good title. We need a good axis. We need our axes labeled. We need good intervals and consistent intervals. So we went up by 2s every time on the y-axis. We went up by 20 every time on the x-axis. We need everything labeled. So I guess I said that. So I guess the axes just mean that you need to make sure you have the correct x and y axis, and then the right scale. So in this case, we don't have hella room on the top of the graph. The graph doesn't go all the way up to 30. On the x-axis, we're not going all the way up to 200 because we have a lot of extra space. You want to always maximize the size of your graph. That's what scaling means. So this is a good graph here. I like the title, Effective Temperature on Enzyme Activity. That could basically be your template title in all of these graphs. The effect of whatever you put on your x-axis on whatever you put on your y-axis. There's your title. All right. And I like this point that they make at the bottom. It says that the points may or may not be connected with the line. You don't have to do that, but it's a good idea. Okay. So quickly, I want to show you something that I have assigned on Canvas for you all. It's an assignment in our Unit 3 module. It says Summary of the Biden-Harris Climate Change Plan. So this is going to be an essay that should be no shorter than 250 words and no longer than 600 words. So that's like half a page, between half a page and a page in length. Not much writing at all. The essay should be typed in 12-point Times New Roman font. So I want you all to get in the habit of writing collegiately. And you will almost always have to write in 12-point Times New Roman font. Make sure your name is on there somewhere. You might even want to have a title. If you want to go above and beyond, that'll be great. I do need to go ahead and put a rubric here. So you guys will see me update that before you submit it. But I provide some links here that could be helpful to you all as you write this essay. Essentially, what you're going to do is summarize President-elect Biden and Vice President-elect Harris's climate change plan. All of these links will be helpful to you, but you're more than welcome to use your own links, to use your own sources, as long as you are citing your sources in APA format. I do provide a link here to help you all do that. If you just go to this website, you basically just have to put in the URL that you're using, and it'll create the citation for you. So again, work smarter, not harder. This is how you finesse in college writing. But you still need to make sure that what you're writing is substantive. You need to make sure that what you're writing is cohesive and eloquent, and that'll be that. Okay, so this will be due on Thursday morning. So we don't have school tomorrow. You all have almost 24 hours, almost 48 hours to work on it. The first time you submit, it'll just be based on that, it'll be based on your original writing, 75 points based on your original writing. Then I'm gonna have you all read each other's writing and make suggestions. And so the second 25 points of the assignment will be based on your edits, but also on the suggestions that you make to one another. All right, so does that make sense? Are there any questions about that assignment? Okay.
No.
All right, well, it's 9:18, so I'm 3 minutes over. I appreciate you all staying. Thanks, Selena. I hope you enjoy your day off, and I'll talk to you on Thursday.